Wait till I show them to you and you'll see why.
Ginormous.
Well, it's what they are is they literally make a slit. So they're not pretty.
So, and like, is the spacing on that one? And then how far on the side?
It depends, so with older tissue,
Yeah.
I need it a little farther back, so you think about maybe eyes,
Yeah.
or the genitals, any sniff or an older animal.
Why is it you can feel the air conditioning when it's
Anytime
like I feel the air, you know?
Last night I was driving home and I thought, oh my God, I've got to turn down the AC.
Yeah.
What is going on? It was.
But they're like full tight down there.
Yeah, no, I don't see any puckering there, and your line of feel is good.
Yeah, I was so excited about this.
Oh, I'm glad you had a good time.
Oh, no, so I'm gonna take a couple different kinds.
Please do, and
There's like with that.
Lish, do you need a banana?
Good things.
I don't want your surgery banana.
Lonnie ages prematurely.
Now, where does it say what the needle size is? 'Cause like, the one I used had one size, had this size needle, and then I stole yours that you already opened, so I could finish, because I ran out
So
of.
this is an FFG.
Okay.
And so, and then this is other respects, so this is the shape of the needle.
Hi, Cammie, can you hear me?
So if you have an actual needle driver for, if you work with the same kind of needle, you would be able to have access to
Yeah, I can hear you.
All right, thank you. Just checking. A lot of my documents are shared in Google Drive, and there's one of them, I'll go over it, but there's one of them, it's the virtual blood for when kids don't want to do it. You have to pay for it, but I'm going to show it today, but I'm not able to share it because I had to pay for it. And it's like $5. So I'll share it for their parents will and she won't let you do it. So I do virtual. So they do, and they have a fake blood lab, but I don't like it very much, you know. All right.
goes into surgery.
Sounds good. Thank you.
You.
So we need to, we should sit for someone who doesn't have a surgery closet and not have one. That's just a scalpel.
If you guys want to try to get around one table, so it'll make it easier, sorry.
Yeah, she did find out about it.
Yeah.
It almost looks fake.
Doesn't it?
We got a creamy napkins.
Like a creamy, the taxidermed animal. Like, that'd be so creepy taxidermed. You need to be extra. My taxidermed.
You're good. Thank you. My aunt like runs our hospital at
Oh,
town, and she had given me like when I was in high school, bags of sutures and
Yeah.
random things, and I
Well, you're gonna need one of those.
need to get it out. And I'm using it now.
Nice.
I have suture pads.
Oh, yeah, I love
Well,
those.
let's put it this way. If you
How
don't
long do
wanna
yours
book
last?
yourself,
I haven't gotten.
you don't have to.
I haven't used them yet. I now have, this year I got another order on Amazon, so I think I have 8 of them now.
Yes.
Do you ever let kids work together, or do you have enough for a whole class?
I have 14, so I think that would be like.
Okay. Okay.
But I sent about last year, with a couple of kids that
But do you remember these
like
things?
needed.
Yeah,
I had this in seventh grade, literally, it's like their anxiety calming.
Well, now this
Yeah,
right
they'll
here,
do
Okay.
it.
Perfect, yeah.
You don't need to be a vet, you could be a plastic
Yeah.
surgeon. Times the amount.
I don't remember my kids. They're kids.
What?
That's why I have a lot of drugs.
Don't matter.
If I do it-
As long as you don't care if sheep steps on you.
If my husband came with me, would that be
Oh, that's
okay?
Yeah.
Okay.
Yep. Then you can have dinner on the way home and it's nice.
Yeah, okay. Thank you.
And then, yeah, I'll go through all that stuff.
I like the ones that are. Do you want a banana?
We'll see.
I'm good. I'm not a banana fan.
I am not really either. I will if I'm desperate, but I'm not.
A banana in a banana split, okay.
Fair.
Banana and candy, great.
Oh, I'm anti-banana.
Really, I love banana candy.
You can have mine whenever I eat candy. I like banana candy.
Because some people.
Runts, or, yeah, runts,
Well, see, I haven't.
Laffy Taffy, holy shit, love it.
Yeah.
And I didn't communicate this, we kind of got screwed up. That ran around that galvanized one, and
This is
those
nothing.
boxes and those little coffee things. And my intention.
No,
was-
the swap rooms. She's in the swap
And we still got those left.
She's in San Juan, I
One
think. So that's the absolute opposite side where we're at.
Toolbox. I don't know if you guys have seen.
That 14-mile trek back.
When I do it, I show it.
Yeah.
So that's kind of, and I think there's enough room for the coffee.
So is that?
And that's just kind of not as part of this now.
Okay, Ashman?
That one has
So-
two, yeah. I know it's true, you
That one planter box?
No, no, no.
And the body has got the toolbox.
Well, there's some old rusty toolboxes. And there's some old rusty, coffee cans in there too. But yeah, that's,
Are we giving 10 shots?
No.
got yourself.
What time is it? Are you doing?
All right, so you would like.
11:04.
Okay, let's go ahead and get started. If you're here for the blood typing lab, you can sit here at this table. I'm sure Cammie can help me. In the Google Drive, I dropped all of my worksheets I'm gonna go over with you guys. And I do a pre-worksheet. The first one is the ABO blood types. It's a super simple one. I have enough of these here.
Yeah.
And it's pretty self-explanatory. And it goes through and helps them learn about the antigens and different types of blood. And it takes and goes, do you want to share that with her? You can put it up on the screen. Let's go to Alt tab. It's this one right here. That's one mouse.
Yeah.
So now we need to go to my Zoom and share that.
The only thing.
Kind of blind, Margaret?
Is it that one?
That works right.
Yeah, it is. I'm having trouble. Okay, this is the key to the one I gave you guys. And basically, it tells you all the answers in the paragraphs at the top. It tells you all the things you need to look at. And then the chart down here in the middle, it helps you answer questions of what one. So it tells you the cell antigen, the red blood cell antigen. It tells you the antibodies, so it doesn't have those. And then it tells you the blood types it can receive from. And then it tells you which ones they can donate to. And it goes through and it basically explains all the information you're gonna see that you need to know. Like if they're typing you and you're in for an emergency, they're gonna donate you, they're just gonna give you whole blood. And then with dogs, if they have a universal donor, they'll do the same thing, or if you're cat. But in humans, they like to try to, if they have the time, they like to blood match you because O's hard to get. So they'll blood match you. They'll usually, if it's a big surgery, have you come in early, they'll type you and they'll have it on hand if they can get it. Same thing with your vets. I do in my vet science, I watch a lot of the videos with the kids, Heartland Vet, some stuff from Dr. Cole. I'm really picky about what I watch with him. But when they show the blood typing, it goes through the process and it shows, and it'll show the surgeries and they show how to do the infusions. And that's why I talk about this, because it's really important, because if they have that adverse reaction, you can lose them real quick. And that's why I go through this. And then it also has a little Punnett square kind of thing where you put it, you go together and it will tell you which blood type and which antibodies and things like that. And so it hits a little bit on a Punnett square, but it's not really a Punnett square because it's a 9, but it helps them understand how to go through this. So this is the first thing I cover when I go through this unit. And then you will have students that will not do this lab. Can you go to the one that shows the pictures? It's on the Google that shows the pictures of the different kits. Because the kit I have, it comes with 2 boxes. They're about $127. It says you can only test 50, but if you can get a Lancet that you can change out that isn't like these here, because they only give you 50 of these ones. But you get enough to where you can do almost 150 kits, but they don't give you. What's that?
Just pass it.
Oh yeah, if you want to use a razor blade and lose your job, you can do that. But you've got to make sure you know the kids take them, throw them away. I forgot my sharps container, so we're gonna throw them in the bottle over there. when we're done. So this is the kit. It comes in 2 boxes. There's not a lot of instructions they give you. If you haven't done this, you have to go to their website or you have to find it online because it doesn't really tell you how to do it. Well, this is from Flynn Scientific. So it comes and you'll have 2 boxes and basically it'll have 2 of these. One will have the concentrated and it comes in a powder. And so with that powder, it tells you use 5 mils of distilled water. If you don't have distilled water, you can use regular, but they recommend distilled. You take and pop it off. You put your 5 mils of water in there. You let it sit like it tells you to. And then after a little while, it tells you to shake and check and make sure it's all dissolved. And then it's done. And then they give you a little beaker. It's got a screw-on cap. I always put which one it is so you know. But they're all different colors. And if you're good, you'll remember. So you do all that, you reconstitute it, and then they give you a little dropper. And that's what you do. And you have that. So the thing's all ready to go. I'm telling you right now, if you have kids that are squeamish, and you need to make sure that they're sitting down when they do this, because I've had kids fall when they saw their blood, and it wasn't one of their buddies' blood next to them. They're like, it's funny to me, but it's not funny when you have to take and lay them on the floor. So you have the lancet. In your kit, you'll get 50 of the lancets. So everybody come grab one, please, real quick. Grab a blood typing card. They do not come laminated. The reason I laminated these is because when they have just the paper, I've noticed over the years that I've been doing this, that basically what ends up happening is you put that blood on that paper card and it sort of starts to absorb and it makes it harder to do the test. So that's why I've done that. And then when you're done, all I do is I use these wipes and I wipe them down. And then I throw the wipes in the garbage because then I take the garbage out myself and get rid of it. Because it has blood on it, but I'm not going to put it in a sharps container and I don't have biohazard bags. So I wipe it off every time and then I'll actually use Clorox. When they bring it up, I'll put it in the tub.
All right.
And then I'll Clorox them myself.
This
So
one.
they're clean. And so you guys, my other science teacher that's not CTE, basically, she's always done paper. She told me, I want you to try it like this. And she did one and she liked it, but we haven't tried it on a whole group of kids. So you guys are my guinea pig set to see. It doesn't affect, because I tried it and it worked. So you're going to end up. And each one of the droppers I label as well, so the kids do not, and I keep these at different tables so the kids can't mess them up. I'm assuming you guys are good enough that you don't. Please grab a Band-Aid as well and grab an alcohol swab. If there's not enough Band-Aids, I'm sorry, I left them in my box at school, because when you jab yourself with these, a lot of the time the kids will set it on their fingers like this here. You can't just set and push because it starts to hurt. You gotta poke it. And some kids are going to bleed like stuck hogs, and some it's going to be really hard. And so you want one of these for each. You need a little stir. So grab one of these so you each have one of those.
We need one of each.
You need one of each color. You do not use them to mix the other ones, because then you can cross-contaminate. So just one from each one. So you need a red, a blue, and a yellow. Yeah, there's 3 ones, because you mix one with the red for your A, you mix one for the B, and then I always use the blue for the red, yellow, and the red is for the RH. And it doesn't matter, but you just got to do it. So make sure you have 3 different colors, please. Uh-oh. This person's not getting to see everything we're doing here. Margaret, can you show her everything you grab when you get it all, please? Sorry about that, Cammie. She'll show you everything she's grabbing.
So I got these little stir sticks. I got a yellow, a blue, and a red.
Okay. So
And I got a
basically,
lance
I strongly advise you guys.
and a
sit
blood
down.
typing card and an alcohol
And if
pad.
you don't think you
and a
can jab
Band-Aid.
yourself hard enough, you might have to have somebody, and this is what you guys need to do when you go to do this lab, your kids, you want to get their fingers and their hands as warm as possible. Because you need an eraser size sample on each of the circles. And I'm talking about, no, like the tip of an eraser, you know what I mean? Yeah, well, like a drop, yeah.
Which finger are we doing, or?
Whichever one you want to poke.
Is there a spot that's?
Just not your dominant hand. So don't do this yet, because I want to walk you through the process so you guys know what you need to do, because as soon as you get your blood on there, you're gonna need to get up here, you're gonna need to grab your dropper, open your container, and start dropping it on there. You do your A, then you do your B. So what I'm gonna do is I'm gonna keep these. The A is gonna be on this table over here.
Wait, so you're gonna take your bloody card with you?
Yeah, because you can. Yes, no, look, because if you pick it up like this here,
Yeah.
and just, these will be out of the way. Just pick it up like this, you bring it people here, and you just set it down. I'm gonna put A here, B here, and see, it's all gonna be at one table. So it's all gonna be at one table, so you're
Okay.
just gonna slide it. And please make sure you put it on the right hand side. There's B.
Thank you.
So he's setting up these dropper bottles with some blue fluid, some yellow fluid, and some clear fluid,
Okay.
which are the antigens, the A and B antigens and the
Okay,
RH
so
factor.
this is what I tell you to do with your students. When you go to start this, you have them start shaking their hand. Get it warm, shake it.
My finger already hurts.
And then you're gonna you milk your finger down. The one you're gonna poke, yeah, sorry. I thought I was teaching teachers, not high school students. And then you take and do that there. And you'll see it gets really red, that's what you want. You're gonna take this, you're gonna poke, and then you're gonna start milking and start putting the blood on each one. Bring your things with you and then start going through. It usually starts clotting, but you guys are supposed to do this till I'm done with the instructions. So once you poke,
We're teachers.
and you've done
We're
that,
gonna do stupid stuff.
you need to take and go over and put your drops in. You use each stick to stir. I only do about 10 seconds stir and then get rid of the stick, but it takes 30 seconds. The RH takes the longest. It can take up to 2 to 3 minutes. They say 5, but it doesn't clot within 2 to 3. That antigen's not there, so you're either A, B, or if none of them clot, you're O. But now if A and B don't clot and the RH does, you're O positive. If all 3 don't clot, you're O negative. So this is how it works. If A clots, you're an A. If A and B clot, you're AB. If only B clots, you're B. None clot, you're O negative. If neither A or B clot, but RH does, you're O negative. And if you're O positive or O negative, your blood's worth a lot of money. And another one, AB positive is also another really good blood type to have. So is everybody clear with the instructions of what you guys need to do? So this is your A, this is your B, this is your RH. So remember, each one has, A is here, B is here, and RH is here.
A is blue,
All right?
B is yellow, RH is
about
clear.
30 kids. This will take you only about 10 minutes. It's getting through the instructions and making sure they're not jackassing around. Because basically, I tell the kids, I said, if you poke somebody with this, I said, you've now just committed a crime of assault. Because if you poked yourself and say you didn't know you had something, you can give them hepatitis. And I say, you cannot screw around. And it's one of those things that some of your districts will not let you do this lab because of bloodborne path. And they're worried about, like right now, COVID, my district lets me do whatever I want, which is kind of surprising. Yes, in the Google Drive, I have my form. I'm sorry, can you pop that up real quick, please, Margaret?
Yes, I can try.
It's a basic form. And I explain to the kids and I tell the parents, if you want to ask what we're going to do, send me an email and I'll call you. I'll talk to the parents. It's something that a lot of kids really like to do, but some kids just,
Which form?
I didn't put my permission form in that.
He didn't do it.
Okay, well, here. Do you have the hard copy? No, that's not mine. Where, it's gotta be in the pile, Margaret. Oh, crap. Did I not do it? I had one with me. I had like.
I saw it.
Oh, no, bad teacher. Oh,
Lonnie failed.
I'm a failure. And in your kit, the only thing they give you guys is this little thing here. They don't give you a lot of instructions. So if you guys have questions, I can answer those for you. But basically all it says is what we're gonna do in the lab that day. And if they don't want their kids to do it, then they don't bring it back signed. And then I give the kids can you pop up that one, hit Alt B for me real quick, please. Why is it not working? No. I had it open. Where'd it go? It's the one. Where's blood typing? No. Okay, that one there. Share that one, please, Margaret. Okay, up there. Those are the top kit is the one that I get. It comes with 2 boxes. It's $127. But when it's all said and done, it gets shipped to you. It's about 150 bucks. And then there's the fake one. If you want to get the fake one, I don't do it. But if you have kids that don't want to do it and you don't want to do the paper assignment, which is a forensics, and it goes through and it makes them follow through the forensics and figure out what it is, you can spend the 46 bucks and do it. So you have a fake one and you have a real one. This is the real one. The fake one's way cheaper, but I think it's more important for the kids to learn the lab process and go through it. Plus it gives them information. And obviously they can't take the cards home because there's bloodborne paths on it.
We're getting ready, Denise, to do our blood samples.
Like sandwich bags, and they can take them home and they dry out and you're fine. But we don't let them take them home anymore at all, just in case. And that's why we're going to try to go to this because it worked for me, so that's why I do it. And so that one, and then we'll go over the assignment, which is the one that you have to pay the $5 to get it online. And it's well worth it because it actually adds another, even if they do this one, I make them do the one online as an assignment. So that's what you're going to do. So does anybody have any questions on what you guys need to do? And there are Band-Aids here. I only had so many, sorry. I forgot them at home because this is not, it's not a small hole and you're going to bleed. Some of you might be like mine. I bled through 2 Band-Aids because I bleed sometimes. I'm not a hemophiliac, but I tore it open pretty hard because I hit it too hard. So yeah, don't have me do it because some of the kids, like some of your students are going to ask you to do it. Make sure you glove up and you change your gloves between each student. Don't stab one and go to the next, and so on and so forth. So if you guys are ready, everything's set up over there. Remember, about 30 seconds with each circle. Oh, sorry. I'm making sure you can hear me. 30 seconds with each circle, and then go to the next one, 30 seconds. Go to the next one, one drop of blood, 1 drop from the pipette. So, any questions?
Okay, so we're doing this.
Does anybody want to be the demonstrator for Cammie at home?
Yeah.
Okay, so here.
We've got Denise now too with us.
Oh, Denise?
Yep.
Hi, Denise. Sorry. So, if you want to go here. So, Did she hear all the instructions of what I do?
I think so.
Okay, so go ahead, Holly. Here, hold on, Holly.
Okay.
Yes, I did. I heard all the instructions. Thanks, and thank you for volunteering so I can see.
Absolutely. Is it on this camera? Okay. So I've never stabbed myself with one of these before, so I'm trying this. Okay. It's kind of nerve wracking because he says you can't just push. You kind of got to. Dude, my hands are shaking doing this. How did I get myself good enough? I got myself really high. I'm going to try it again. This is hard to like stab it. My hands are shaking. How much coffee did I have today?
Oh,
I stabbed myself like 15 times. So have your kids work on their aim. And then you kind of milk it, is what he said.
Well, most people can't push it in because it doesn't feel the best. Like, see, that's the second time, like this one here.
Oh, geez, Will Pete. Got blood everywhere. Just lick it off. That's really classy, Holly.
That's here. You gotta get a lot.
oh, good God. What is she doing?
I was trying to milk it and it slipped off and I just flung blood in my face.
This is what you need.
Lonnie,
What?
I just flung blood into my face.
Hey, Holly's up here trying to recreate a murder mystery.
I think I need a different finger. There we go.
God, you're just whacking it.
Well, he's like, stab it. I think,
Doing a great job, Holly. Better than I would have done. So
does it have to like drop or can it like be like,
You just gotta get a droplet on there.
these things suck.
Well,
I
don't,
think
yeah,
they're that great.
don't drop it. You want to get your finger and just like dip it on there. There you go. And where's your poker at? Here,
Yeah,
I trust
sorry,
you. I
sorry.
trust you.
Yeah, my blood's all the time.
Margaret, did you do it yet? Oh no, you got it. You got to get. You
Is
got
that
to
enough
like she
blood?
does. You got to get it on there, otherwise it won't work. You might have to poke.
Now I'm
this
gonna
up
go do
again.
that stirring and I'll come back and show you. You can figure out what blood type I am.
Okay, so that's not enough. You look at Holly's really quick. That's what you're looking for. And that's what, oh, you stabbed yourself 3 times?
In one finger and then like another one.
And then she gushed it all over the air.
No, I didn't. That's why I wasn't watching it. Because see, if you get it right, you can keep going like this here. You should be able to pump out blood like this every time. I did the side. Don't do right here. Do the side. Just like if you're doing a blood test for diabetics, you do it on the side because you bleed, and then you can take it like this here. And you just take it like this, and you can just and see that's enough right there. That's if you got that'll work. Sweet. Always got enough. See, that should be enough. If you need a little more, you can. Are you gonna solve them now?
You shouldn't have said anything. You should have just let us do it like blue like
Oh,
this.
whatever you guys do,
Yeah.
you
don't tell
don't do
people
that.
everything.
Do the side.
I did that.
You get, but some of you guys are gonna have the same problem I do. You
No,
work with
she's
your
gonna
hands,
answer
your
this.
hands are super callous, and it's hard to get it out.
And then it stings all over the table when you stir it. You're gonna be like, how did those are
I know, I probably. Yes, I did.
And I did like a drop
Here.
in a high chair.
Is that enough?
I try to get more. Yours is perfect. That's good. Put all your samples on there. Get your samples filled, and then you just do one at a time. Mix it, and then just let it set. And do the next one. See, that's perfect right there. You can just put that on there. You're perfect.
It wasn't that bad.
That's good. Now put another one on there.
I was trying to post my video, but I get it on my face.
They just mix it for 30 seconds.
Here, I think you just have that down.
Okay, make rough it like that.
So you're using your friends.
Because I was going to use this for the dump.
I feel like I didn't have enough blood.
Oh, you got it.
Okay. So I'm angry. See, when I do that, it wins it. I think I put too much. I'm just a high sugar pressure. Yeah.
Yeah, she's A.
Yeah, it's like.
It does actually, it's my mom has to work. And her daughter actually. Like it lets relation. And it's funny, she doesn't drink, so she'll try to make
So if it does not, it's negative.
Yeah, that means you don't have to answer. So you are A.
That's safe to the kids.
And that's what you are.
I'm an A flat.
Yeah, I'm an A B positive, which is, it's a good one.
I'm pretty sure that's orange, right?
The RH one is.
Yes.
And then, yeah, one is orange, just one little drop. That's all you get.
I'm still trying to get enough blood out.
I'm sorry.
Is that enough for this?
Oh, yeah, your first 2, you're ready. So you guys just, like the kids, I make it jab. I make the kids jab because if you try to do like that, most kids will not sit there and let you go back and forth. You just got to push it in.
I ended up A negative.
They start feeling it, they say nope.
But I kind of like.
Thanks so much, Margaret, for all your narration and showing me stuff. I appreciate it.
And Holly's over there working. She's A positive, but she's just like, "Ugh." She's really
Yeah, I can only imagine.
flinging blood all over. I think she's coming back now.
All right, so when you guys are done, I don't want to tip it too much. You should see that on one of your sock sections, it's coagulated. And depending on what coagulates, turns colors, whatever, is going to, here, I'll put this down even more. There we go. It's going to determine your blood typing. There you go. Can you see that? So I am A positive because this guy didn't, well, it's starting to clot. I don't know. I'll have to ask him, but according to this, I'm A positive.
Thanks for sharing, Holly.
You're welcome.
So wait, this doesn't count as cardio,
So, yeah, take one of those, wipe it all down. You have a what? Well, you're gonna dump it in that copy thing, then I'm gonna take and I'll keep it shut, so. And if you have to use 2 of those, I don't really care. So then those ones there, I'll just have you drop in the box.
So on the little card, we wiped them all down with alcohol wipes. Hey guys, in Cornell, they've got one of these with milk and vinegar and water.
So it makes it to where it clumps when it.
Yeah, and then they actually, this bird got killed and you have to figure out who the killer is. And it's one of the lady's pets. It's kind of cute. Ziggy's the killer. Ziggy.
Can you pull up the are
Your
you a killer?
battery's running out.
Jesus.
Your battery's running low, so I hope we don't lose you.
You have
He's got
power
the power
here?
cord. Yeah, there's a power strip right underneath this corner.
Thank you, Margaret. So that's A. No, you're A negative.
So what do we bring up?
It didn't coagulate. If it was coagulating, it'd be fine.
Okay, because I'm like, I know I'm A negative, but Holly said positive, so this means negative
You're
because
A,
it
you're
didn't.
A, so you're A negative.
Okay, that's what I thought, okay.
Okay, so then you grab a wipe, drop it in there. Okay, so is there anybody else that's gonna try to do this? Because I want to get through the one assignment I go through that has a forensics part.
Okay, which one is it?
It's so go to the Google Drive.
I gotta go back.
Oh, it did.
What the heck's going on? Why is my drive not opening?
It doesn't, it's positive.
Fine, I'm gonna cheat. Oh no, we'll go back.
If it does,
So back here?
wait, how do you tell if it coagulates? It turns color, it looks color.
Wait, you said I was A positive. She says if it coagulates, then you're
You, that's,
negative. If just the A coagulates.
if A coagulates, you're A. If B coagulates, you're AB. And if the RH coagulates, it makes you a positive.
Oh,
If
so
it
I'm an A.
coagulates, you're a positive.
Oh, you told me I was an A positive, and that didn't.
I thought you were RH clotted.
Did it?
I thought
asked
it
you if
did.
it clotted, and you're like, that doesn't look like it clotted.
So we'll just show them the answer one. So just slide it. Yeah, if you go through just do the slideshow thing. Yeah.
And so it might not have got mixed, and that's sometimes, but it's been with, I do my kids in the 4 years I've done this, it's been about 98% accurate. So sometimes it makes mistakes. The ones you guys have used, just take and make a pile right here. You guys didn't use Band-Aids, so you must not bleed that bad. I'm still kind of bleeding. So up on the screen, this is the lab that I have kids do that I make them all do. I'll get this up there, let's see. I'll put it all away later. It's a forensics lab. So basically, the first slide, it shows you, this is the RH antigen. So if it has the yellow square on it, that tells you it's the RH antigen. If it has the blue bubble, that's the B antigen. And then if it has the green triangle, that means it's the A antigen. So the next slide, because I'm going to give you guys the one that has all the answers, it tells you, it says antigens on the surface of the red blood cell. So A positive, you go back to the last one, you look at the diagram, unless you can remember. You remember the A antigen would be present, and the RH antigen, because it's positive, would be present. And then, so that means the antibody it does not have is anti-B. It's really self-explanatory. So that would be the first slide they go through. Then they go to the next one. And so it says, a person with type B blood or B negative blood, red blood cells looks like this. It tells you to bring the stuff over that you're supposed to do. So this one here, you put the circles on and you drag your answers out of this box over here and you attach them. And you attach it to make it look like a person with B negative blood type. And so it shows you how to do it and you go over and you drag it. So grab it, drag it across. Well, you should be able to drag it across. Is it not working?
No.
Okay, when you go into Google Drive from the share, it will make it to where you do it. This is the answer key, so it might not be doing it. So you drag it across, you drop it there, and you connect it. And that makes you your A negative because the RH isn't on there. So you go to the next slide. So this one here, you can see where they grab these and start sticking them on. That's what they're going to do. You're going to drag it from the other slide and start putting it on here. You're like this one here, it's only going to be that one. This one here, none of them attached. This one here, this is an A positive. So there's your A and there's your RH positive. And that's what it does. So you drag them from the slide before in because these won't be here. So you want to have them drag those in from the other slide and put them on here. And then you can start moving them around and putting them like they're supposed to be. Oh, thanks, Holly. So then you go to the next one. And this is.
Hold on. There we go.
This is the one where it talks about your crime scene. Do not cross. This is where the CSI part starts. So basically it talks about it's early Saturday. It goes through, somebody was killed, blah, blah, blah. And it gives you the scenario. And then it goes to the next slide. And as you read through, it's going to give you two suspects.
Hold on, I'm getting there.
So you have Mrs. Brown as suspect number 1, Mrs. Gray as suspect number 2. And so you know these are your suspects. And then your task is basically to perform a blood test and find out who the victim's head belongs to, of the two suspects. So it gives you your kit. So there's your antibody. It shows you this. You play the video. It'll show you which one clumps. And then you Go ahead and show the video, Margaret.
Oh, okay.
Sorry, it's not very loud.
It's somebody whistling.
Well, you're not gonna go dun, dun, dun. Okay. So what they do is they show this, and it shows you the date. Obviously, it's there. You're gonna drop the one, drop the one, drop the one. And this will actually take a period. It'll take them about 50 minutes to get through. So they're using the fake blood test. And now they're gonna take, that's the fake blood, now they use the antigens and they put it in there. No, this is the key. I'm showing you the key.
Okay.
So once this is all done, it says, did it have anti-A? It's yes. Anti-B, no. Anti-RH, no. So that's what it does. So now they're going to show you guys the different ones. We don't have the white ones, we have red, yellow ones. So you start bringing it across, and you start, I just drop it straight in and start doing it. You see now how it's starting to clump? It's not staying like that gelatinous. You know, it's not staying like fluid. So that one's a positive, but it's clumping. Our blood, you don't see ours be like quite like that. It'll be like always, which is really obvious. It gets thick and super like, we do a CMT test. It starts getting thicker and then the more somatic cells it has. So you can see negatives, you can see negatives. So from that, they would say yes, anti-A, no, no. So go ahead and take this one off. This is Olson.
Okay.
And then you go to the next slide. And it says, looking at this anti-A, that's a question of blood sample. So basically, they watch the video and look at it and go to the next one. So it talks about what they see. They would say which one it is. Here are your possibilities, and you'd pick the right one. So this narrows it down to possibilities to either A or
Come on.
A positive, which we know it's not A positive. So then what you do is you drop it down to the next slide. And it tells you that it's A negative because you know you don't have that square antibody for the Rh. And so you know it's A negative because only if you make them write this, how did they determine? It makes them think and expand on why they came to the conclusion why it was A negative. They have to explain that the B did not coagulate, so the antigen is not there. The Rh did not coagulate, so they are a negative. So you make them explain that. So it makes them think, and they have to explain their answer to you. They have to explain their argument. So this is the one where you play the next one. And the thing is, this is the second suspect. This is suspect number 2. Lo and behold, she's A, and anti-B yes, and Rh no. So it's AB negative. So let's see this one.
No, we're good.
You found the victim's head was tight match from suspect number 1, Mrs. Brown, but the blood found near the victim head of Miss Brown was A negative and Miss Gray was AB negative.
What happened? I did.
Go back one, please.
Way to own it, Margaret.
So
Thank you.
blood types form the class evidence that's A negative. So in reality, there is not enough to convict because there could be a possibility of it being contaminated or crossed. So they don't have it. And a lot of the kids will just rush through and say it was the first person because it came across. But you have that second one there, and there's not enough information to technically convict because it's circumstantial evidence. And you could argue, if you're a good criminal attorney, you could argue that the second person, it could have gotten contaminated, and it's not enough. And this is the answer key. So,
But I do DNA.
hey, I don't know what to tell you. I'm just saying, if you
I would do
blood type-
DNA. I'm
you-
watching
they
enough
would go to DNA.
NCIS. I know this. I'm giving Abby the blood samples. We're nailing that bastard.
Okay. So basically, that's what I do. I do the worksheet. Then if they do, and like, I like making the kids do the forensics one, even if they do it, because it helps them go through, and when they're going through the process, thinking about and coming up with an argument as why they came up with that answer. That's why I do it. So on the Google Drive that they're going to share out, there'll be a thing that has the web page that you can go to. It's called Teachers Sharing with Teachers. Everything they have on there is like $5. I have another one I'm doing tomorrow. It's a genetic Punnett squares, and you breed dragons, and then you do Punnett squares with it, and you can actually build a dragon. If you have two kids, one breeds it in his, another one breeds it. You make them partners. They bring their genetics down, and they make their own dragon and mix it together, and you show the offspring. And it's a fun one. That one can take up to 2 days, depending on your kids, because your slower kids will start getting. If they don't understand Punnett squares, like I have a process when I go through Punnett squares. I'll go through a video they watch that's timestamped. Then I go to the next one where the assignment will actually go all green if they get the right letters in the squares, and if they get the right phenotypic and genotypic expression. And so it goes through, and then they understand how to do a simple Punnett square. And then when you get to the dragon one, it's really simple for them to do it. And that's a $5 lab you can get too. I wish we could find one that they had cows or pigs or something, but I'm not smart enough to figure out how to do it. So
Yeah,
I stole it.
Denise Lonnie talked about that a little bit, and his school lets him do whatever. Mine, I think wouldn't, but I think I might use sheep blood because they're okay with that, but they're not okay with human blood. But there's also the fake one that you can do, this online one. And there's also another one on the Cornell that actually uses milk and vinegar and water. And that's on the Cornell vet curriculum, and it's another option that you could do. So it really does depend on what your risk management lets them do. Every school is different.
Thanks so much. Yeah, I don't know if I would do it with my own kids. I think if it was animal samples, that would be a better one.
And animal blood does the same clotting factors, so it's pretty straightforward.
I hope this was helpful. Yeah, those ones weren't used.
Okay, I think he's done. You guys have any more questions?
Thank you very much. I appreciate it, Margaret.
Yep, no problem. Talk to you later.
You can do the fake one. I used to like. I used to do the milk one where you use milk and vinegar. Same process, but I like making it think, it's human or your animal. And you have to make sure you know, because if you're wrong, you give that blood to a dog or a cat or whatever you give a horse, off it goes and it's done.
You think first of all, if it has to work, if you put it in a sample tube with some EDTA, that should still be able to factor. And we all have access to some type of animal.
Yeah.
I think I'd do it with my sheepies because I do blood smears with my sheep.
Yeah.
So that's why that's right. So I'm A positive.
It's really positive.
Okay.
Because see how it's got the little.
Yeah, I
you're
didn't see
positive.
it before, but now it's
Yeah.
showing up. It showed
See,
up
it's A positive.
and I didn't wait long enough.
So thank you guys for listening to my spiel. Hopefully I didn't bore you too much. And if you guys have any questions, or you can shoot me an email, call me anytime. If you're having trouble, like if your district doesn't want to let you do it, I'll give you the websites and where to find it. But it's all on Flynn Scientific.
Thank you.
You're welcome. Thank you.
Everything's back.
Hey, I ended it for you, Lonnie.
What's that?
I ended it.
Thank you very much, Margaret. Much appreciated.
No problem.
But I don't want to set this table because you lead on it. Oh, I got this table.
Holly, you cleaned out each one of the droppers and put them back in the
Yeah, I
same.
rinsed them out.
Okay, you put them back in the same.
Yeah, everything's back in there.
That's, I just put them in there just so I don't lose them.
More extra here, but you have these here.
Well, we have engineering.
Right, to make sure that it's, I should, college biology, and so I. Yeah, yeah.
Now, did you have to make that waiting another month?
They used yeah, 2001, when I was doing that. Yes. But, I think. Like, I mean, the like, what diagnetics use, like the.
So that's why I.
I have to look back and double check on my supplies, because I've got my own books that I have. Yeah, exactly. Well, and that's why I was around this table with that. I jumped myself like that a little bit, and then I couldn't. Yeah, like the first time I thought it got good, and then the second. That's yeah, exactly, and that's what I would do.
But then once they got their pivot, it's
And I also have type one, so I walk. Super fast. So. Yeah. Yeah.
All right, there you guys. I hope it helped you.
I want to do it. I drove around yesterday.
Should have been, what the heck?